everybody. Thank you very much for joining. Good morning, good afternoon, because we have people from several places. So this is Luis Balino. I'm International Area Manager for BioArray Lab. So again, thank you for joining. I hope this presentation by our lab colleagues, our PGS, PGD specialists, will be interesting for you. So well, I will not say anymore. Just let our colleague, Dr. Vanessa Penacho, introduce herself and start with the presentation. Please enjoy. At the end of the presentation, please feel free to make any questions. The best way is using the chat. You know, this system includes a chat, so you can type any questions there. Thank you and enjoy. Please, Vanessa, all is yours.
Good morning, good afternoon to everybody. Thank you for your assistance to this webinar. My name is Vanessa Penacho. I'm the R&D responsible here at BioArray. As you know, our company is situated in Elche, Alicante. And now I want to present you our results about how next generation sequencing platform, the PGM by Author, has improved us the detection of chromosomal abnormalities in early human embryos. Well, my company is a technology-based firm specialized in genetic analysis through leading technologies in molecular biology, mainly array-based techniques and NGS, both applied to clinical and research purposes. We cover preconception, preimplantation, prenatal, and postnatal diagnosis. Nowadays, women gain the right to control our own fertility. However, this freedom of choice has consequences due to the impact of lifestyle factors on reproductive performance that undergoes infertility. The apparent fecundability is decreasing in women after 35 years of age. Now, to get down to the business, I would like to point out some basic notions. As all of you know, ploidy is the number of sets of chromosomes in cell nucleus. A human gamete, has a simple copy of each chromosome, so a normal human cell has two complete haploid sets. Therefore, we are diploids. This is a perfect situation, but this is not always like that. A chromosome anomaly, abnormality, or aberration reflects on a non-typical number of chromosomes or a structural abnormality in one or more chromosomes. These anomalies are present in at least 10% of sperm and 25% of oocytes, and suppose around 50% of spontaneous first trimester miscarriages. We define aneuploidy as an extra copy or absence of a chromosome, and this is the core of PGS, the preimplantation genetic screening. There are two main mechanisms leading to numerical chromosome anomalies that happens in meiosis 1 or 2 or in mitosis. These anomalies are due to a non-disjunction in homologous chromosomes or sister chromatids. Mitotic non-disjunction results in somatic mosaicism. Well, as I told you before, one important factor of aneuploidy is the maternal age. The birth frequency of chromosomal disorder increases with ages, especially after 35 years of age, at the same time that the implantation rate are decreasing. The risk of aneuploidy in a live-born child is 1 in 192 at advanced age. Also, the proportion of embryos presenting double or even triple or multiple aneuploidies is greater with this increasing maternal age. So, in these cases, in vitro fertilization and genetic analysis are strictly recommended. We are always talking about maternal age and women's fertility, but what about the importance of other age to disease risk? The rate of de novo mutation, especially related to autism or even schizophrenia, increases with the age of father at conception of child. Other important situation that compromises fertility and requires genetic analysis is the presence of structural chromosome anomalies. Translocations are caused by rearrangement of parts between non-homologous chromosomes. In balanced translocation, there is no exchange of material, so there is not going to be extra or missing genetic information, and chromosomes have full functionality. However, carrying one of these translocations produces unbalanced products in embryos and increases the risk of recurrent miscarriages. Also, another case study is represented by monogenic genetic disorders, which are the result of a single defective gene and are inheritable, such as cystic fibrosis, Huntington disease, or Fragile X syndrome, for example. What about the in vitro fertilization? Every cycle of IVF involves several steps: ovulation induction, both oocyte and sperm retrieval, fertilization, genetic analysis, and finally the embryo transfer. A genetic analysis supposes always PGS, the pre-implantation genetic screening, that checks embryo for numerical chromosome abnormalities. This is known as aneuploidy screening, but in some cases, PGD, known as pre-implantation genetic diagnosis, checks future parents for structural chromosome anomalies or single gene defects of unaffected embryos. Well, in IVF, embryo biopsy and subsequent genetic analysis can be performed at the time embryo fertilization has been checked out. But mainly, biopsy is carried out at day 3 after fertilization. This is known as blastomer biopsy, or at day 5 to 6. This is known as trophectoderm biopsy. Blastomer biopsy is more traumatic and decreases the embryo's potential for implantation. However, during trophectoderm biopsy, more cells are removed, so trauma to the embryo is minimized. Day 5 biopsy coupled to PGDs and PGD increases implantation success rate. Many studies have shown that aneuploidy screening of day 5 embryos increases that implantation rate compared to those studies based on morphology alone. Lately, this screening has been assessed by ACGH, DRI-based technology. PGS by ACGH is fast. It has a protocol and also an analysis easy to handle and requires a simple equipment, but is quite expensive. Nowadays, next-generation sequencing, NGS technology, supposes an alternative tool for evaluating multiple genomic loci, chromosomes, and samples at once. Its quality, robustness, and cost-effectiveness offer us important advantages over microarrays technology. Even semiconductor sequencing on PGM platform, Even semiconductor sequencing on PGM platform by Ion Torrent allow multiplexing and scalable PGS and PGD. The first workflow on NGS for PGS we validated was the low-pass whole genome aneuploidy one against ACGH. Here, we detected ploidy states of all the embryos with an ultra-low read coverage sequencing across the genome, even less than 0.01x. Here you can see a schema about how the DNA sequence reads are mapped to each chromosome interval that we know as beam. The more extensive is the coverage provided over the whole genome, subdivided into these chromosomal beams, the more precise is going to be the prediction of ploidy states. About results of this first validation phase, we take 147 embryos, 144 day 5, and 39 day 3 biopsies. Day 5 embryos present a concordant screening between NGS and ACGH that suppose around 97.96, around 98%. Day 3 embryos with the concordant screening were 100%. So this PGS by NGS suppose less money and less time for us. Here you can see a result for a euploid embryo and normal embryo analyzed by both NGS and ACGH. Top graph shows NGS analysis and bottom graph shows ACGH analysis. Here you can see comparison of both techniques for an aneuploid embryo. And also here you can see an embryo with unbalanced translocation due to parental inheritance. Also, in this first validation study, we look for the minimum number of reads necessary to provide reliable copy number variation of the ploidy stage of each embryo. We conclude that 50,000 reads ensure a map threshold of 0.3, significant enough, but this copy number quality control metric. Also, for mosaicism study, we mix reads for aneuploid and euploid embryo in order to generate different and normal embryos with different percentage of mosaicism. Regarding to each chromosome site in general, we conclude that NGS approach detect, we conclude that this NGS approach detects a mosaic level in a proportion of 20 to 80. It supposes around 20% of aneuploid reads to 80% of euploid reads. Also, we compare mitochondrial DNA content of euploid and aneuploid embryos analyzed by NGS. Despite other authors that have found significant difference, after analyzing more than 1,300 embryos, we conclude that euploid and aneuploid embryos are identical population at 0.05 significant level regarding to chromosome M ratio. This is calculated as mitochondrial read counts related to autosomal read counts per embryo. Sorry for this graph because it has a problem. After low-pass whole genome aneuploidy screening, the second NGS workflow that we validated for PGS was the ESHRE. With this new approach, we decreased time in template preparation with isothermal amplification and also in the final analysis. With ion reporter software, we can now visualize many embryos analysis at once, summary of gains and losses, and other parameters. And also we can manage filter options. Also, this new workflow gives us an accurate screening in a balanced translocation products due to parental inheritance. About results of this second validation phase that compares second NGS workflow to the first one and also both against ACGH, say you that we tested 20 embryos biopsied in triplicate, and we got an absolute concordance between the three techniques. This new PGS by any NGS supposes even lesser prices and take us shorter time. But we don't stop. We are always considering new challenges, so we need to spend even less money and get results earlier. So we decide to couple PGS to PGD in order to achieve results from both chromosomes and gene analysis and at the same time. We prepare PGS and PGD libraries simultaneously, and we pull libraries with a proportion of 1 for PGS to 6 to 10 for PGDs. So we have increased the number of flows to 500 along sequencing, which allow us to get enough coverage for PGS and PGD. Combination of ultra coverage sequencing for PGS of around 0.01-fold and high coverage dip for PGD of around 20-fold give us a complete study of embryo analysis regarding to both aneuploidy screening and genomic region involved by single gene mutation. Going further, PGD must be also coupled to a previous informative testing. Single nucleotide polymorphism markers linked to this gene region involved by mutation are evaluated in order to avoid a possible misdiagnosis due to the well-known allele dropout effect. We have developed and implemented in our lab a high-throughput tag SNP genotype combined with ion ampli-seq sequencing for the identification of point mutation, ensuring enough informativity. The LD-TAC SNP tool from the National Institute of Environmental Science in US provides us a linkage disequilibrium for around 130 SNPs selected within each gene. This tool are preparing us the DNA sequence for primate design by AmpliSeq, considering all polymorphism information in 200 kilobases flanking regions at both sites of point mutation. Here you can see an example of PGD analysis by NGS for neurofibromatosis type 2. Male was carrier of a point mutation in NF2 gene, and we can prove that two resulting embryos, the ED2 and 3, presented also the mutation. Here you can see how we perform informativity study by SNPs for the same case study. We could also discard the embryo 1 due to an informative SNP linked to mutation. Finally, this third validation study has been performed in collaboration with 5 institutes studying many point mutation and carrier couples. We have analyzed by PGS a total of 4,718 embryos with around 98% concordance to previous technique approaches we used before in our lab. For the moment, around 15% of TAC SNP started were fully informative, but we are increasing this rate right now. The overall LN-Duproud rate was less than 3%. With 1,238 embryos transferred, we have an implantation rate around 60%. Finally, we got the cheapest and the fastest protocol. Just to sum up with all the advantages that the NGS platform, PGM by Ion Torrent and Thermo Fisher Scientific, provide us in pre-implantation studies, say that it's fast, it's adaptable, is scalable, especially if we compare to other NGS platforms. We can use different chips depending on number of samples multiplexes. Also allows us to combine PGS, PGD, and informativity studies. And finally, is cost-effective and cost-efficient. Just to thank to all my team here at the BioRaves lab, and thank you so much for the attention and your patience.
Thank you very much, Vanessa. So, please, do you have any questions? Any of you for Vanessa? As I said, you can easily type in the chat the questions. Okay. Well, if no questions, then please remember, keep in mind that we will be at ESHRE, both in the exhibition. We have a booth, number A55, where we will receive all our clients and future cooperation IVF centers. And also we are bringing there a couple of research works, precisely on, of course, on PGS doing NGS, PGS and PGD. So we have 2 posters there. There you will be able to meet Vanessa and Luis Alcaraz as well, our lab director. Okay.